To close out the month, we have a fantastic lecturer. I've had an opportunity to see him give several talks at conferences, and it is always enjoyable. Not to far too high.
Yeah, no, whoa.
But we have Dr. Lee Murfee, who is professor and senior associate chair for undergrad studies at the J. Creighton Pruitt Family Department of Biomedical Engineering at the University of Florida, which of course is just up the road. And Dr. Murfee joined the University of Florida, I believe, after starting his faculty position at Tulane University in New Orleans. He had completed his bachelor's in mechanical engineering at MIT and his Ph.D. in biomedical engineering at the University of Virginia, and then a postdoctoral fellowship at UCSD. He is the past president of the Microcirculatory Society and has a lot of interactions, I know, with Dr. Sukias through some of his work in that realm, and current president of Alpha Eta Mu Beta, the editor-in-chief of Microcirculation, and a fellow of BMES, AAMBE, and the American Physiological Society Cardiovascular section. His research is focused on scientific discoveries and bioengineering approaches that utilize both in vitro and in vivo models and computational approaches to explain angiogenesis and vascular patterning, lymph angiogenesis and neurogenesis to describe a variety of different cardiovascular and neurovascular function and dysfunction. And I know that in addition to research, he's very passionate about education. I read that he has won the Departmental Teaching Award 4 times, I believe, at the University of Florida, which is outstanding, and he is a big proponent of empowering students and promoting critical thinking in our student population. So with that, I would like to welcome
Okay, Josh, thank you very much. That was super nice. For me, the first take-home lesson for today is I like to think I haven't changed since grad school. So I don't know what I've been doing for 20, 30 years. That sounds good. But really, I haven't changed much since grad school. And most of the ideas in what I'll share with you today were actually motivated by questions when I was in graduate school. And I remember going and asking my PI, who was some big name in the field at the time, and even other faculty that worked in our labs. And I remember them not knowing the answers. So I learned very quickly that sometimes the PIs don't know the answers, which is okay. That means I'm not going to know all the answers today to the questions that motivate the work and that that motivate what I'll show. All right. So that's my first message. The second message is really I'm okay being interrupted. Anything goes. You just feel free to butt in. And my question for y'all is, how many are PhD students? Okay, and how many are undergrads? Okay, great. All right, let's get into it. So I'm going to try to, what we do in our lab, the pointer doesn't work there, is we look at cells involved in blood vessel growth. And Josh also mentioned lymphatic vessel growth. And that's what I'll highlight today. And I'll put it in the context of a couple different stories. For those of you who don't study blood vessel or lymphatic vessel growth, what you can try to appreciate is the same cell types I'll talk about show up almost in any tissue. So whether it's atherosclerosis, whether it's valve repair, it's still the same cells dancing around doing stuff. Big picture, if I were to describe what we do, we try to look at this real time at everything that's on the left-hand side of the screen. So blood capillaries, lymphatic capillaries, and all the cells that dance around in the tissue space around it that coordinate to influence growth of either system. So what I'm going to share with you is kind of our views and how we do it. And the big picture questions that we might think about are, well, can we develop new views, new models to lead to consideration of what we know? I mentioned in grad school I learned that PIs don't know everything. I also have learned that sometimes when you read something in a paper, it doesn't mean the people writing it know everything also. So I'm going to give an example of trying to make sense of the literature. And then we'll spend more time on giving an example to the second question, which is, can models lead to discoveries? What I really like doing, aside from other stuff, is being under the microscope and looking at cells and trying to, my happy place is thinking that no one else is looking at this stuff when we look at it. Don't know if that's true, but that's what we like to do in our life. So, all right, let's get going. So, for the most of the talk, I'm gonna focus on a tissue culture model that we developed. But I'm gonna take about 5 minutes and introduce some computational work that we do in our lab and with a collaborator that considers shear stress and angiogenesis. So I'm just gonna jump into it. Shear stress is thought to be a regulator of angiogenesis. Okay. It's most of the studies in the literature that paradigm comes from actually has to do with shear stress effect on endothelial cell behavior, and it's actually motivated by atherosclerosis. So a couple years ago, we looked in the literature, and I'll explain this. This is the percentage of studies, and we took the first 100 studies we could find in the literature on PubMed. So percentage of studies on the y-axis. On the x-axis is what shear stress value they used. Most of these studies are in vitro. And you can think of most of them are either in a parallel plate flow chamber, or a microfluidic device. But the key is to understand what's going on, and we're going to come back to this, is most shear stress values are 0 to 30. Okay, so what you need to remember is 0 to 30 shear stress stimulating endothelial cells, and that's what people think or base their statements and review articles that are related to shear stress being involved in angiogenesis. All right, so I'm going to give an example at the network level. Y'all can imagine what a network of a microvascular network looks like. It has arterioles, venules, capillaries, and lymphatic vessels. That's what's important as we move to the next slide. And I'm going to try to share with you an example of using computational modeling based on what you would learn in a fluid dynamics class your junior or senior year here to gain insights. So on the left is a view of a mesenteric microvascular network. So this is connective tissue that's off the gut. This is images taken from the rat. You can actually, the connective tissue is similar to what's in humans. The point is the yellow are perfused vessels. And there happens to be an angiogenic network, and that's supported by really high density kind of regions of networks. Right. Let's move to the right though. So if you take the image on the left and you just apply Purcell's law for each vessel segment, okay, and you use MATLAB, you can get to what's on the right, which is an estimation of shear stress per each dot is a segment. So in this network, there's thousands of segments. The take-home message, though, is most of the shear stress, well, one, there's a big range from 0 to 1,000. Don't know if 1,000 is physiological. Actually, I'm not sure if it's not physiological, but there's a big range. Okay. If you're thinking back a couple slides, most of the experiments in the literature are 0 to 50. So let me show that heterogeneity a different way. This is just a color map, and each color maps to shear stress values. So I'm just going to take one column, and I'll take this one. So these are different zoomed in regions of the network. And actually, the bottom two rows show the same thing. They're just different y-axes. So let's go on down to the bottom here. The scale is 0 to 50, which is what most of the experiments in vitro do. And you can just see there's different colors. Blue, red, okay, orange, yellow. So all that means, and what you're supposed to appreciate is we go blue right next to a red. 0 right next to a greater than 50. And again, some of these shear stresses are hundreds of dynes per centimeter squared. Right? So that was two-dimensional. You could think, well, is that physiological relevant? Right? It's really hard to measure shear stress in an animal. Actually, you can't measure shear stress. It's really hard even to measure what you what you need to estimate shear stress. So we, for a couple years now, we collaborate with a friend at New Jersey Institute of Technology. He's in mechanical engineering, but he develops these really cool 3D red blood cell resolve simulations. What's important for today is that on the left is just red blood cells flowing in a network. This is in silico, so in computational model. Much fancier than the model we use, which is what's based on what you learn in fluid dynamics. This on the left looks kind of like what you see in vivo. On the right is wall shear stress distribution. So if you're looking at it, just pick a segment. And I'm going to pick this one. And the red just has to do with hotspots of shear stress. Okay, and you can imagine this is affected by the red blood cells flowing through the networks. All right, but what I want to leave you with, though, is red, blue, something in the middle. So in the tissue space, we are one segment away from each other, and we're changing colors. And that's the same thing we showed with the simple 2D model. And what it just highlights again is that each vessel is experiencing totally different shear stress values. So we're less important with what the exact number that we get with our models. What we're more interested in is the literature in the ballpark. And at least with these two different approaches, let's go back to the literature. There's plenty of examples of shear stress being estimated to be in this range, which happens to be not in the range that most of the literature is focused on. So in this first part of the talk, what I'm left with is, well, what I kind of thought when I was a grad student, which is I'm really not sure how to make sense of the literature. And I'm not sure what the real shear stress magnitudes are. And what's fun for me when thinking about research is, well, this is not a definitive answer, but it contributes to the scientific discussion and contributes to at least my understanding of when I go to a conference, when I see a bigwig talking and they go, this is the schematic everyone needs to follow. It's not always that straightforward. Right, that's my computational work. Now I'm going to shift. In the last 2/3 of the talk, I'm going to get back to what our lab spends most of our time on, and that is trying to look at what's on the left-hand side of the screen in real time underneath the microscope. And we do it to try to understand what cells are involved in blood vessel growth, lymphatic vessel growth. Where do cells come from? Where do cells go? And I'm going to start by introducing the whole motivation. And I would say our lab is right at the intersection of physiology and engineering. So a tissue engineering challenge, probably something that that resonates with you, is how do we mimic complexity of a real tissue? Okay. Do y'all take tissue engineering classes? All right. So you probably learn about assays that have to do with, or approaches with, cells and a scaffold. You go to a biomedical engineering conference. Has anyone been to BMES? Some people. Okay, you go to BMES. There's awesome rows, almost like a sea of posters, focused on how do we get cells happy in a 3D scaffold. Sometimes it's multiple cell types. Sometimes it's the scaffold. Sometimes it's, well, I'll leave it at that. Sometimes it's about biomaterials approach. Sometimes it's coming from the cell side. The goal is to match the in vivo complexity. And this is not for tissue replacement. This is for learning about basic science discovery and drug testing. So probably about 10 years ago, we said, all right, we want to get here. We don't know how to get the schematic that I showed you on the previous slide, the 20 different cell types, the patterning. We don't know how to do it. And we can come back to discuss if people are close or not. But the point is we started something new. And it was new for our lab. It happens to be still new with regards to the researchers we hang out with in our field. But it's not that new as far as an approach. It's just culturing a tissue. We didn't go very far. We cultured the mesentery tissue. So we can culture tissue from the rat, tissue from the mouse. We're starting to explore culturing tissue from humans. But the point is we cut out a tissue and put it in a dish. And the reason is because we want to view those 20 different cell types in the patterns that make sense and mimic in vivo. Now, my disclaimer is any model, any approach has its issues. So we got our own issues. But for us, it allows us to look at certain things that other models don't allow us to look at. All right, so that sets up what I want to talk about the rest of the talk, or the rest of my time. I'm going to spend a few slides and describe the model, and I'll overview very broadly kind of what the capabilities are, and then I'm going to present a discovery that we made that we still attack in our lab, which has to do with lymphatic vessels turning into blood vessels. I'll describe it as we go through it. Then I'm going to end by linking that to the tumor microenvironment, so cancer, and getting back to in vivo, which is where my lab started, which is really the whole goal of what we're trying to learn about, which is dynamics in vivo. And along the way, I'll try to make a few points. Okay, let me introduce our model. I already said it's simple. It's really simple. So I'll focus on the left here. Here's our mesentery tissue. If you open up a rat, there's about 20 to 25 of these tissues that you can cut out. And you can put them in a Coulter dish. You can actually buy little inserts and smush it down and keep it flat. So, right. I'll remind you, the goal is to see something like this underneath a microscope. When I was in grad school, what I was impressed by is like, oh, the really cool labs have movies. So then I was like, I got to get movies. So I'm going to show you one or two movies. That's it today. And this is one of them. This is a tissue harvested from a GFP rat. And it's a good reminder. The thing I like about research is you get to decide what your inputs are. So the introduction sounds nice. My goal was to get movies. That was my goal. So today I'm not going to show you all the movies, but I'm going to show you a couple. So this is the important thing is this is GFP rat tissue. So every cell is supposed to be GFP positive. The more important thing is you can see the blood vessels. I'm going to play the movie. This is in a dish. I'm going to gloss over the details, but this is in a dish for about 3 days. So you can focus here or here. Let me play it. And you can see like a bloom of vessels, hopefully. So here it looks a lot crazier than when it started. Let me play it again. Okay, so the good thing about this tissue is you can zoom in at different vessels and cells. And so we can zoom at muscle cells, parasites. Parasites are a cell we like to study in our lab. They are, if you don't know about them, they are cells that wrap around the endothelium along capillaries, the smallest vessels. You can also see down here, it's green lymphatics, red blood vessels. You can see these networks of vessels. And I'll come back to that, but go ahead.
And this is in a static condition?
So, okay, these images are static. You can imagine, this is...
There's no flow through those.
No flow, yep. You can remind me, after the talk, I can show you, we can introduce flow. And we've done that. It turns out, just like for any model, we normally don't do it, because for some questions, we don't need to do it. And it looks easy when we show you the result, but it just takes a long time to do in the lab.
Yeah, speaking of time, what time frame are we talking here?
Okay, the movie's about 3 days.
3 days.
Yep. And we took images, it's a stack of images, of course, and we probably took images about every 8 hours. Yeah, we might have gone 12 hours overnight, not sure. Okay, so I'm gonna get the lymphatic vessels and blood vessels interacting. We have 2 main projects in our lab. One has to do with that, and the other has to do with looking at stem cell fate. So, all right, so just stay with me. I'm going to jump to our interest in stem cells. So we've always been interested in where stem cells go and can we form new vessels. So going back to when I started grad school, it's about the time that people started injecting stem cells and people getting excited about them incorporating into the vasculature. All the excitement was them becoming vessels or cells along vessels. Okay, that shifted to, well, maybe that doesn't happen and they just secrete stuff. The image here is an image of red adult human stem cells. This is from bone marrow. And we put them onto our rat tissues. Okay. This is probably 3 days later. I forget if it's 3 days or 5 days. But I want to focus on two things. One is these clustering of cells, and the other are cells lining up along vessels. And this is what we care about, is can we increase this? I would argue that most people inject stem cells, and then you look in sections, there's not that many views of that would allow you to gain insight into where they go within a tissue. Okay, so I want to come back to this. Can we form vessels? And what we did years ago is we teamed up with a stem cell lab, and they had different populations of adult and age stem cells from ASC, adipose-derived sources, and bone marrow-derived sources. The important thing is if we quantify the percentage of cells in that parasite location, which is what's shown here in the arrows, wrapping along the small blood vessels, the range of percentages across our groups of stem cells was 0 to 10. So you can see the averages. This happens to be the percentage that maps to what people report in vivo. It happens to be what people have reported for the last 20 years. It really hasn't changed. There's a couple exceptions to that, but that's still, I feel confident in what the range is. Okay, so then we moved to Florida. Josh mentioned I was at Tulane for 10 years, then I moved to Florida. Research is interesting. My Ph.D. was at UVA. There was a plastic surgeon at UVA when I was there. I moved to Tulane, I moved to Florida. He was at Florida when I was at Florida. Okay, we teamed up and started collaborating. He was interested in SVF. Has anyone, stromal vascular fraction, has anyone heard of this? So stromal vascular fraction is adipose tissue, so fat tissue minus the fat cells. It has all the fat stem cells in there, but it also has all the stromal, all the other cells in the tissue, including endothelial cells and other vascular cells. Okay, so it turns out what plastic surgeons do is they inject SVF and similar types of slurries of cells into patients. This is really big in breast reconstruction surgery and, let's say, flap or wound repair. They don't know where they go. Our interest is, can they form vessels? I showed you on the previous slide, injecting stem cells, you get about 0 to 10% that contribute to new vessels. What you don't get is totally new vessels. All right, so now let me go to this slide. The GFP, and what we see here, is the host network. So these are host vessels in the tissue. So these are vessels that are already in our mesenteric tissue in our culture dish. We put this SVF onto the tissue, and I want you to focus on this no vessel region. And I'll toggle back and forth. So after about 3 days, the no vessel region fills in with vessel segments. And I'm glossing over the details. We label the vessels, all the vessels in this case, so we still see the host vessels, but we see these new vessels filling in. So for us, this happened about 4 or 5 years ago, and now we are focused on SVF, and the reason is, as far as our stem cell slurry population, and the reason is because it has this dramatic effect on growing new vessels. More so than I've seen with any individual stem cell population. And we're trying to figure out, are the vessels functional? Do they connect? What the connections look like with the host vasculature. But this type of discovery kind of is what our lab's about. Now it's a jumping off point to figure out what's going on. All right, so let me get back to what we're gonna talk about the rest of the way here, which is lymphatic blood vessel plasticity. Here's my last movie. That's it. But this is, for me, this is one of my favorites. Let me explain it. So the lectin label is just a label that identifies endothelial cells. It does a little more, but for today, it identifies bright blood vessels, and then these gray ones, can you see the gray ones? Those are the lymphatic vessels. This is before remodeling. There's a gap here and a gap here. I'll play the movie twice. So the first time you can focus on one of them, one of the gaps. The second time, just switch your view to the other gaps. Okay, so the top gap gets filled in. We'll look at the bottom gap here. And that's the in position. It's filled in. There's a gap, and then it's filled in. We'll play it again. So I would argue, it sounds super simple. We're just looking underneath the microscope, and we're seeing this. And it's really just made possible by trying to look at things in a new way. In our case, it's blood vessels and lymphatic vessels at the same time. So now I guess it's, for a while, I go to lymphatic conferences and I go to blood vessel conferences. Most of the lymphatic researchers used to study blood vessels. It's not common that I see images of both vessel types at the same time. So all right, we'll play one more. So now I'm going to focus on this image and the after. So the before and after, we can see it's filled in. And what we've done here is we've taken the tissues and then labeled them immunohistochemically. So we've labeled for PCAM and live 1. Live 1 is the lymphatic marker. So all I can tell you to add context to this image is this vessel here is typically, or in the before, I know, based on experience, is 100% red. And now I go here and I look, segment that's green, no red. I have red. Okay, the red becomes choppy. I go down here, green, no red. So somehow, all these red cells are becoming non-red. So there's some phenotypic switch. And we know because we watch over time that the lymphatic endothelial cells remain. Actually, if you really paid attention, this segment came from the lymphatic vessel. And now it's green and no red. In the lymphatic world, lymphatic endothelial cells are lymphatic endothelial cells because they have the red label. So that's quantified here. And what you see after this remodeling, when you look at both vessel types and you're able to view them over time, is a decrease in the percent coverage of lymphatic vessels with lymphatic marker. So going back to the previous slide, it's a decrease in the amount of red label along the lymphatic segment. So before remodeling, it's 100% red. Afterwards, you get these kind of mosaic looking images where you get some red and then you get some breaks. That breaks translates to a decrease in the coverage. So something's going on. And this motivates additional questions, which are, well, what are the regulators of this potential lymphatic blood vessel plasticity? And for our lab, does this relate to any pathology? It turns out this lymphatic blood vessel plasticity, the first time I showed it without the movie, years ago, it was half the audience at the conference I was at said, no way, we don't believe it. And then what's funny is they had a poster session after the talk and it involved some wine. And then there was a lot more people saying, no, they don't believe it. But, okay. And then they're like, you need to show, of course, a movie to convince us. So then I came back to a movie. And since then, there's a couple more examples from the literature to show that this plasticity can happen. But still, there's just a handful that are out there. So as far as pathological conditions, I'm going to jump to tumor microenvironment. Turns out the tumors, yep, go ahead.
Just to clarify, you're saying that in the literature, what most people believe is that these two never interlink.
That's
Lymphatic
correct.
vessels and blood vessels never connect.
Yep. If you pick up a physiology textbook, the connections happen at the thoracic duct, the big vessels. And then the other areas of connections, which is not really connections, but there's openings in the shared space in a lymph node. So, but at the capillary level, it's thought that they're disconnected. So we don't know yet what the functional implications are. All right, so I'm going to go to tumors, and for different reasons, we start we asked a simple question: is do these lymphatic connections or examples of plasticity transition from lymphatic vessels to blood vessels happen in a tumor microenvironment? To do it, we wanted to use something close to our model. So it turns out, based on our experience, we knew if we put something on our tissue that whatever we put on could integrate. So the model is just really simple tissue culture, and then we added tumor spheroids. And we just went upstairs to a lab that made tumor spheroids, and we started collaborating, and we put tumor spheroids onto our tissue. Let's take a look. This is what it looks like under the microscope. The red here is di-eye. That's just a membrane label if you're not familiar with it. And we see two spheroid structures. So these are two spheroids. And the green is our lectin, and it identifies lymphatic vessels and blood vessels. The important thing here, in the cancer cells, if it matters to you, is H1299. This is a lung metastatic human, very common cancer cell line. So the point here is that we can see underneath the microscope our spheroids and where lymphatic vessels and blood vessels are. All right. So let's focus on what's on the right. On the right is, we're on the edge of our spheroid. Okay. We're using this lectin, the green, to identify our blood vessels and lymphatic vessels. We know lymphatic vessels based on going outside this view to seeing what the morphology or shape of the vessels look like. So I can tell you, you can take my word for it for today, but is that this vessel is a lymphatic vessel. Okay, this is actually a blood vessel. And we can see signs of connections. Here's another example. This is a lymphatic vessel, this is a blood vessel. This little hot spot is something's going on. There's some interaction, which we call connection. We also know going to the right, in this case the spheroid is a little outside the region shown here, is that we see evidence of lymphatic sprouting. We published a paper a year and a half ago or so that just established that these tumor spheroids on this tissue resulted in characteristics of what's in the literature regarding blood vessels in the tumor, which is increased permeability, increased angiogenesis. The new part was also looking at these vessel connections in mosaic vessels that I mentioned. So let me explain what the graphs are. What's shown here is control tissues. This is no spheroids and spheroids. And there's an increase in the number of connections. What the images show here is our control scenario, which is minimal essential media only, and then tissues with our spheroids on them. In a control tissue, it looks like what we would see in vivo, which is green blood vessels, red lymphatic vessels, very few connections between them. In the tumor spheroid tissue, we see connections, but more importantly, we see these gaps. And going back to our movie, because it's about the movie set, going back to the movie, what we learned was these gaps come from, we learned what the gaps mean. And what the gaps represent are these lymphatic vessels kind of transitioning into blood vessels. And all jokes aside, the reason that's important, if we just see this, we don't know where the cells came from, we don't know. So now we're putting studies together to make sense of the observations that we see here, and we really think this is lymphatic vessels that are turning into blood vessels. Something's going on. Okay, so remember, for later, which is just a couple slides from now, is that the cancer cells can cause lymphatic vessel plasticity, and they cause these gaps, and the gaps have to do with loss of lymphatic identity. So, I mentioned our lab, we think physiologists and engineers. So for Nick and I, one of our home societies, the Microcirculatory Society, okay, not that big of a deal. But what it is a mix between physiologists and engineers. So I don't know about you, Nick, but for me, I go to conferences and half the people I'm presenting to are physiologists. So if we're presenting computational work or tissue engineering motivated work, we're trying to convince physiologists that it mattered. So this is different than going to a BMES and just talking to engineers. And it's a little more of a pep rally. Like, hey, great job. So, all right. Do you agree, Nick? Yeah. So sometimes the physiologists are really critical. All right. So I knew for us, the first question that anyone asks is, well, so what does this matter in vivo? A challenge for tissue engineering models, and we can discuss this later, is in vivo relevance, so physiological relevance. The challenge is really amplified when you try to show it. And usually what labs do is they compare it to in vivo scenario. With that though, the scenarios are totally different. Think of like a microfluidic device. Okay, think of just cells in a dish, and then you're going to compare what the cells look like or their phenotypic, what they express, and you're comparing it to a real tumor environment. It's just not the same. So, but that's kind of as good as we can get. So we tried to do something, and I don't think this is perfect, but we tried to compare as similar scenarios as we can. So I described the ex vivo tissue culture model. What we did in vivo is chronic in vivo experiment, we take the tissue out, and I knew we could do this because I used to do it in grad school. And what I would play around with at grad school, it didn't go in my dissertation, but as I would add stuff to the tissue. In this case, we're adding tumor spheroids. And then we take the tissue after a few minutes and put it back into the animal, and then we wait 3 or 5 days. So I'm going to show you what some of the results look like. Okay, and I'm going to try to explain, can you see this? It's like a big, kind of weird looking thing. I'll try to explain what this looks like. And what I can tell you is that we only see this when we see tumor spheroids. So we're still learning. So here is evidence that tumor spheroids remain on the tissues. So the dye is labeling our tumor spheroids. You can see I said it's not perfect. The cells aren't as clumped as we noticed in the culture, so they spread out a bit. But day three, 2 different examples at different magnifications. This is the whole tissue. This is zoomed in. We can see some clustering. The far right image is red cancer cells, and the PCAM is labeling our vessels. This tissue is not live. We cut it out and then we label it. The dye we use, if you've used it, it says CM here. It's a fixable dye. So that works pretty well. But the point is cancer cells stay on the tissue, and we can see blood vessels and lymphatic vessels. This is in vivo, so our chronic animal experiment. What we also learned is, check the first row out. Here's the junctional labeling. This is no spheroids. Okay, let's look here. The label is PCAM, so endothelial cell junctional marker. You can see outlines of endothelial cells. Looks like puzzle pieces. Here, there's no outlines. Actually, I was pretty shocked by this. So on the bottom is the green. Let's forget about the red now. The red has to do with cancer cells presence, but the green has to do with the presence of a dextrin that we injected through the femoral artery. So this is something that was in the vessels. Okay, let's go to the right. This case, very few no spheroids put onto this tissue. So don't know if cancer cells are on there or not, but no spheroids. You can see what the green does. It stays within the vessels. In almost every tissue that we looked at that had tumor spheroids, the green was all over the place. So, when we looked at tissues zoomed in, and we could find examples in tissue that had spheroids where the green was, there's little hot spots of leakage. But most of the tissues look like this, right? So this is important because it mimics something that goes on in a real tumor environment. This is a real tissue environment, but we're just putting tumor spheroids on the tissue. We don't know. We don't know if it matches all the characteristics of a real tumor. Right? So I'll explain this one. Tissues with cancer cells Well, it was hard to find lymphatic vessels. So let me explain. PCAM is our endothelial marker, live 1 is our lymphatic marker. So here's PCAM, it's labeling. So the labeling doesn't look as clear as what we've seen before. In part because the PCAM labeling is being broken up. Also in part because we label these tissues with a different fixative that we normally label. In this case, it's paraformaldehyde. If you fix tissues with paraformaldehyde, it kind of messes with how clear the labels are. But let's focus on the arrows. So PCAM vessel, okay, we know based on morphology is the lymphatic vessel. It's hard to find lymphatic or identify lymphatic labeling. Compared to the unstimulated tissue, we have PCAM, and then you can clearly identify lymphatic vessels. The live one also labels interstitial, probably activated macrophages. But the point here is that you can see these lymphatic structures. So it turns out, when we first started to look at these cancer cells, these tissues with cancer cells, we only were looking at the ones with lymphatic vessels. And we were like, wow, so interesting. So many of our tissues don't have lymphatic vessels. And then it forced us to go back and quantify. What's the percentage of tissues that have lymphatic vessels? And I'll focus on the white and then these two bars. But you go no stimulation and then stimulation. And I can come back and explain the subtlety of these differences or lack of differences. But the point here is that in a remodeling group, even with spheroids, there's a seemingly drop in the tissues that have lymphatic vessels. Okay? So we think lymphatic vessels are being lost. So let me get back to this odd-shaped looking thing. And what I'll tell you is white bar versus black bar, we only see these type of structures with our cancer cell groups. And we don't know what they are yet, but they happen off of lymphatic vessels. So we call them blebs, but the blebs they protrude off of lymphatic vessels. And here's an example of a smaller one. Here's a PCAM label, so we know they're endothelial cells. And if we look at live 1, there's part of this structure that actually doesn't label for live 1. So not only are they protruding off lymphatic vessels, but they also are losing some of that lymphatic marker, which is consistent with the phenomenon that I introduced before about lymphatic vessels turning into blood vessels.
I have a question.
Yep.
Are these spheroids, like, in other models, like injected to cause cancer in
Okay,
animals?
good question. So in tissue engineering models, sometimes if you're familiar with the common microfluidic devices, they have a channel and a channel, and then they have space in between. So they'll put the spheroids in the space in between. That's one thing. Okay, 2, for in vivo, there's a handful of studies that have injected cells IP, and then harvested and then found out or harvested mesentery later to look at the effect on the mesentery tissue. And in some cases, people have injected spheroids. So there are examples of people using something similar to this approach to mimic or study a tumor microenvironment. Okay, the only add-on comment says these are lung metastatic cells, and a big question we get is, well, is that relevant? Our goal here is mapping back to the tissue culture. Our goal in the tissue culture experiment is to use almost any cancer cell type. It's just a platform. So the mesentery tissue doesn't matter so much. And it turns out in culture, we can put on breast cancer cells, mouse, lung cancer cells, so we put on multiple cell types and we see similar behavior. So that's a long way to say I don't know how well it maps.
I'm more curious about like the day 5 as well. Like, is that enough? If you left it more time.
Oh, I see. We don't know.
You would be able to see what the flow is.
Yeah, we don't know what this will look like eventually.
Right.
Yeah, that's a good question. Because we've only gone out to day 5. Yep. Okay, with that, I'll try to wrap up and then leave 10 minutes for questions or something. But, all right, well, thank you very much. At least I shared kind of what we do in our lab and how at least the last 2/3 are interested in meeting the challenge in tissue engineering of how do you get something in a dish that looks like in vivo, motivated our tissue culture model. That led to, for today's talk, a discovery of lymphatic blood vessel plasticity. And now we're going back to trying to match it to in vivo to guide what our understanding is regarding whether this can happen in vivo. And what's interesting for us is we'll take our observations and we go back to our chronic animal experiments, but we also go to observations that collaborators have made. And we try to make sense of the observations made in the literature or the observations made by collaborators. So, and if I leave you, I guess I showed you a couple movies, but for our lab, it's about trying to learn stuff. Really, it's stuff that I knew we didn't know when I was in grad school. So it stayed with me over my career so far. The way we do it is to try to look at things slightly different. And what I'll impress upon you is really not complicated. At the end of the day, we just cut a tissue out and culture it. We have undergrads do it, we have grad students, it's really pretty simple. Maybe the skill comes with knowing what you're looking at, which sometimes isn't, well, in our lab, it just means everybody looks under the microscope for a long time. Which goes back to, that's an important skill. Sometimes, yeah, that's not emphasized enough. So, all right, with that, thank you very much. I'm very lucky to have collaborators all over, so, and I'll take questions. Sure.
Hi. Have you guys started looking into why this plasticity might happen? Is it because of like some sort of mechanical stress or like secretions by the cancer cells?
Okay, yeah, for our lab, I would ready to go if I were to take a step back. It's discovery, and then we use that to ask these questions why. So what we're doing in our lab now is we're focusing in on a couple molecules. And there's one in particular, it happens to be a couple labs down from us. And sometimes this is the way research works. There's a lab who studies immune cells and immune modulation. And that's really big in the tumor world. Okay, it turns out the molecule that he's interested in, it's IDO, that's the acronym, IDO, you can look it up, but that is secreted by these tumor cells. And almost nothing is known about its effects on angiogenesis and lymphoangiogenesis. Then it turns out that if we put IDO onto our tissue, we can get these connections. So we don't know the mechanisms yet. For our lab, we're starting to go after one of them. I'm sure there's a lot more than one. And it happens to be a molecule that other people around us are interested in. Sure.
Great talk. I love the whole thing all the way through, honestly. I was just wondering, like, it seems like the lymph nodes themselves kind of like break down a little bit
Yep.
based on the data you showed, like the loss of junctions and like the leakiness of the stuff that you were injecting. Is that like degradation of like blood vessels for angiogenesis, like a thing that happens in the blood vessel side of things?
So for the blood vessels, so the leakiness has to do with the blood vessels.
Oh, okay.
And usually, like in a tumor microenvironment, they're characterized as being very leaky. So, and you can think about it as your junctions just split apart or become looser. So in angiogenesis, if you think about it, you're going from a vessel and then you gotta have cells, so they have to start breaking apart. What happens is you get some junctional breakdown.
So that like normally happens.
So that normally happens, and that's a characteristic of the tumor environment. That happens to be leaky and happens to have lots of blood vessel growth. So, if you were to look at tissue, and let's say one part of the region there's no growth, and one part of the region there's lots of growth, this is the area, this is the region that would be leaky.
Cool. So then that process of breaking down the blood vessels for the purpose of growth, is that something that you think that the lymph nodes are also doing?
Okay.
In like a parallel kind of way?
Yeah, good question. So what we learned is that the lymphatic vessels, the lymphatic endothelial cells can change their identity and become like blood endothelial cells. And what we also learned is an extreme version of that is actually just going away. We don't know what that means as far as like function yet. As far as, do they turn into blood vessels and then, yeah, those vessels are functional and have blood flow. We don't know in our lab yet if that happens.
Awesome, thank you.
Yep, Nick.
Another question. So, should we assume your findings that suggest that there is actually a system that allows the blood vessels to hijack lymphatics in order to accelerate angiogenesis?
Okay, that's a great question, because that could explain why like a rapid angiogenesis effect. So that is logical, and that's what we think could be happening. I don't know if that's important.
If this is the case, what happens to the red blood cells?
The red blood cells.
Yeah,
Yep.
it goes as the lymphatic.
Yes, good question. We have not seen yet, this goes back to your question, if red blood cells can enter the lymphatics. We haven't seen this in our model. There's a couple examples in the literature. There's one, this is the way it goes. So we published in Microcirculation. There's an example, maybe 3 years ago, 4 years ago, that showed in zebrafish, red blood cells going into lymphatic vessels. Okay, that one was published in Nature Medicine, I think, but that's the way it goes.
Go ahead.
Yeah, you mentioned earlier that there are about 25 different types of receptor tissues.
Yep.
Does the type influence the plasticity?
Okay, no, so just to clarify, it's like imagine like a wheel with spokes. There's 25 spaces, so the tissues are all kind of the same, but there's just 25 of these very thin windows that go around. Yeah, and the spokes are big vessels that feed the gut. So there's like 25 tissues that we can cut out and are kind of the same that we can culture.
And does the geography of that in terms of the microenvironment influence how the microbes appears? And to affect like the leakiness of the vessels?
Okay, that's a really good question. I don't think so. The little extra detail there is there's 25. Depending on where you are along the gut, some can have more blood vessels than others. So when we go in, even though I said 25, we try to take the same 8 or so from a specific region. So.
Oh, okay, go ahead.
You mentioned at the beginning of your lecture that shear stress promotes the aggregation of the stem cells to the area of high shear stress. Is that correct?
Okay, yeah, that's a good question. I guess the big message was that shear stress can change per vessel.
Right.
So, in the literature, okay, interesting, there's, when you get a growing vessel, the cells along the vessel are characterized as different types, stalk and tip cells, but different depending on the location along the sprout. What's also in the literature is if you have this shear stress value, that could cause this cell identity. And all we're really getting at, if you were to kind of apply our understanding is, well, that just doesn't quite make sense, because one environment you might have a high shear stress, one you might have a low shear stress. Yeah, does that make sense?
Yeah, but so is it thought that there's some kind of chemical communication to chemotaxis for the stem cell, something like that?
Yeah,
Is it
as
not?
far as stem cells moving or getting recruited, yep, I didn't talk about it, but there are examples of chemotactic gradients and migration
Okay.
of cells.
Thank you.
Just like there is with immune cells being
Right.
recruited to areas of growth.
The lymphatics like kind of, to me, it's kind of like dark arts. Kind of, I don't want to get it. It kind of scares me.
Yeah, lymphatics. Oh, yeah.
Yeah, right. Well, exactly. So I'm wondering, like you mentioned, like functionally, I don't guess it's really you don't understand kind of what's going on there. But like even just the endothelial cells themselves, lymphatic endothelial cells versus the vascular endothelial cells, blood vessel endothelial cells. What, other than the phenotypic markers, what are the differences in how they behave?
Okay. I'll cheat because I'll stay with the markers. For all intents and purposes, like there's 3 markers people use, and it's Ly1, potoplanin, and prox1. That's what they use for lymphatic endothelial cell identity. So, and then well, this is just the way it goes with some research areas. The lymphatic research, for some reason, they were really hot, like in the 80s. Don't know why. Then around, around 2000, vascular people started to give them attention again. So Georg Schonbein's lab was one of the labs that helped or one of the groups. Now, and that started like a Gordon conference for lymphatic vessels. And now, and when it started, it was like whoever studied lymphatic vessels, like 200 people in the world, they went to the lymphatic conference. Now what happens, like in your field, everyone who does studies lymphatic vessels for the most part are related to those initial people. Okay, the reason, this is my long answer, but getting short here, is people are still kind of interested in the same questions. And they're either interested in bigger lymphatic vessels that pump, or small ones, and they're interested in lymphoangiogenesis or immune cell trafficking to those vessels. So as far as the differences, there's not a great answer. You know, someone would say, oh, dendritic cell trafficking, maybe how they interact. You know, but it's not really clear, oh, this lymphatic vessel is different from this. And it's phenotypic, and then it's what people describe as the junctional labeling and like the structure of the cells. So that's my take on it.
But this smooth muscle of the larger lymphatics has more, some cardiac.
Yes, yeah, there's overlap. And so everything I showed you today with lymphatic vessels, they don't have any smooth cells on them. So they're all just endothelial cells.
But the larger lymphatic.
But the larger lymphatics do, and then the people who study them used to study smooth cell contraction. And so now what's happening, if you look in the literature, is what's emerging is the same players are being shown up, and what's being reported is what's similar and what's different. And there's lots of similarities, and there's some difference, because the pumping's different, just like you would have differences in cardiac muscle and smooth muscle, or, yeah.
So you.
Yeah, I had a question that I think would have been nice for undergrad taking transport or cardiovascular mechanics. Again, going back to the beginning, we were talking about the different shear stresses and how we can model them in silico. I was wondering if you could talk a little bit about what assumptions can or can't be made at such tiny scales in the microvasculature, and it kind of a talk we had way back when by Dr. Shelley Weinbaum, who was here talking about the.
Shelley?
Yes.
When was he here?
It's been a few years.
It's been a few
few
years.
years.
Yeah, yeah.
I think pre-
Oh, I love that guy.
His
Yeah.
talk resonated with me because he talked about the glycocalyx and how
Yeah.
it.
And skiing? Did you talk
Yeah,
about?
talking about skiing and a
Train
train.
tried to be trained.
And
Yeah.
then a train. There's a train involved in there somewhere too.
Yeah, he's one of my favorite thinkers. Yeah, go ahead.
Yeah, yeah. So, you know, talking about something like the non-split boundary condition and how
Yeah.
the glycocalyx.
Okay, so, all right, so we're out of time. That's a much bigger discussion.
Yeah.
Okay, but I try to give my real take on it, and Nick, you can chime in, because Nick does more complicated models. So the same model that we use, that we published on, and actually, Nick, you were on one of these papers, is the same model that I used when I was in grad school. It's the same model that one of the leaders, Tim Seacombe or something, uses, and then and it really hasn't changed. Okay, and it's a segmented model, so it's really just applying Poiseuille's flow to each segment. You could argue whether whether viscosity changes, whether you need to think about other stuff. This note, but for our lab, I guess I'm like, well, it only matters if it matters. So the take-home thing was that simple model. The reason we're able to publish with it, if you got me on the spot, so I'm up here today, is because, well, it's really the question we're asking. The question we're asking is shear stress the same in every segment? So you can use that model to get that approximation. Okay, so then I showed the 3D model. The 3D modeling has to do with more complicated red blood cells. Then you're talking about two different fluid behaviors, and actually stuff I don't understand. The take-home message from that is still you can get different shear stresses. So the real simple modeling we do in our lab really strips down, it's as simple as we can get, and it just points us in a direction. Does that make sense? So that's it. So that's a way to kind of to cheat. But yeah, that's my answer. So,
All right, let's
because of
leave.
course it can get more complicated. Go ahead.
Murphy again.
Oh, thanks.
That's how you.
Yeah, really.
real science.
I don't know if we're getting into it.
What's my motivation?
They don't really, no one wants to look at those. They're not bright and shiny, you know.
Narcotics in the brain.
It's my.
So that happens.
It's not a cool thing. I like it.
Actually, you guys did it. It's like,
Amazing, Michael was.